Want to know the truth behind stem cell research? In this video, we're going to dive into the actual scientific paper written by the researchers who figured out how to turn adult human skin cells back into stem cells. Stem cells possess a fascinating capability called pluripotency, or the ability to turn into nearly any cell in the body. The ability to generate stem cells from adult human cells could open up the door to use a patient's own cells to make stem cells match to them, potentially capable of replacing damaged or diseased organs or types of cells. For example, say we had a patient with a severe spinal cord injury resulting in paralysis. Wouldn't it be incredible if we could take cells from their skin, turn them into stem cells, and then use those stem cells to replace the damaged cells in their body, restoring function? But real quick, scienceisyou is a growing channel dedicated to making real biotechnology research available, accessible, and fun for everyone. To support this mission, please subscribe and spread the word. Thanks for watching. Welcome back to this sequel on the discovery of Induced Pluripotent Stem Cells, or iPSCs. If you haven't yet, watch the first episode linked above. Previously, we had discussed a seminal research paper where Yamanaka Factors were identified. These are 4 genes that, when activated, are able to make adult cells turn into pluripotent stem cells, capable of turning into nearly any type of cell in the body, cracking the secret stem cell code. But wait a second, these experiments were all done in mouse cells. Can they be repeated in human cells? Or was this just a biological anomaly? In this paper, the researchers attempted to turn human adult cells into human pluripotent stem cells that have the potential to become any cell. But to recap, here's how the previous study transformed adult mouse cells into mouse stem cells. They injected active copies of these 4 genes into the cells using viruses. Viruses attach to cells and inject their own genetic material, tricking the cell into making more of them. But in the lab, viruses can be made to contain different genetic material, such as active copies of genes, and used like a mini needle and syringe for the cells. So we can just toss the viruses on human cells and get iPSCs, right? In theory, yes. But in experimental biology research, theory and reality often don't quite agree. The thing is, for this viral gene insertion strategy to work, a couple of things have to happen. First, obviously, the viruses have to be able to successfully attach to the cells and inject their genetic material. The virus does this by recognizing and attaching to special features on the cell's surface. Second, the viral genetic material has to successfully sneak into and mix with the cell's DNA, so the cell starts making it. The process of inserting genes we're interested in into cells via viruses is called transduction. Whether or not it works like we wanted is called transduction efficiency. For example, if only 20% of the cells take in the genes we wanted, the transduction efficiency would be 20%, which is pretty low. Unfortunately, this is what the researchers observed when they attempted to insert the 4 genes that make stem cells into adult human skin cells. So they had to improve the transduction efficiency, or in other words, make the virus gene insertion thing work better. An easy way to see whether the viral transduction is working or not is to encode a gene for a fluorescent protein that glows under the microscope into the viral gene that's being delivered, so that it can be easily seen whether or not the cell took up the gene and started making the fluorescent protein. Here, the researchers used GFP, or green fluorescent protein, to observe their transduction efficiency, and from there tried to improve it. But how? So the situation was that they had a virus with their 4 stem cell gene factors packaged in, and it attached well to mouse cells, but not to human cells. Like mentioned earlier, viruses attach to cells by recognizing certain features, particularly proteins on the cell's surface. So the researchers took a different virus that attached well to human cells and used it to put an active copy of the gene for protein that viruses attach to in mice. Think of it like changing the number on your mailbox so that now you get the neighbor's packages. Then, when they applied the virus with their 4 packaged genes, they got a transduction efficiency, or success rate basically, of around 60%, which was much better. So now the 4 gene factors for stem cells were inside the human adult cells. But the most important question, did this turn them into stem cells? And how could they tell? On a petri dish, cells might start out as individual single cells, but then as they divide, they remain attached, forming small groups of cells called colonies. Under the microscope, these colonies look like small islands. These islands or groups of cells often appear different to the eye, depending on what types of cells are there. For example, from far above you could probably tell the difference between a lush tropical island and an Arctic snow-covered island based on just how they look. Similarly, stem cell colonies have a unique appearance. These make round colonies with a smooth edge containing very densely packed cells. This is in contrast to cells that appear more spread out or cobblestone-like, or what the authors describe as a granulated appearance. So even though the viral insertion of the 4 gene factors didn't turn all of the cells into stem cells, the authors could go through and pick out the colonies by eye that appeared to look like stem cell colonies. But of course, they didn't just rely on their eye. They wanted to see what was going on inside of all the cells too. All the cells in your body generally have the same DNA, but your body isn't just one big blob of cells, of course, but made up of lots of different types. These types of cells look and act different from one another, largely because of the types of proteins they're making. In the lab, there are a few ways we can look at the proteins a cell is making. Each protein is encoded for in a section of the DNA called a gene. Before a gene is made into a protein, it gets copied into an intermediate form, RNA. In scientific papers, usually experiments to study protein production look at both RNA and the actual proteins being made. Quantitative PCR is a method of essentially counting how many RNA molecules are in the cell that correspond to a certain gene. On the other hand, Western blotting is a way to get an idea of how many of a certain type of protein are in the cells by basically staining them as dark bands. Using these two methods, the researchers found that the induced stem cells indeed contained RNA and proteins that are characteristic of stem cells. Also, they compared their human-induced pluripotent stem cells, iPSCs, to human embryonic stem cells using a microarray, which looks at a large number of RNA molecules being made by cells, thousands, in order to get a much more comprehensive view of what types of proteins the cells are making and identify patterns or differences between types of cells. They found that their iPSCs were very similar to human embryonic stem cells, but not quite identical. A small number of the genes they examined, around 4%, showed a greater than 5-fold difference in how much protein of that type they were making. That seems like a pretty good match. But remember, the whole point of making these iPSCs is so we can turn them into different types of cells and tissues, right? So the authors next tested whether the iPSCs could differentiate or turn into different types of cells. They found that indeed the cells could be differentiated into several types of tissues. Embryoid bodies, for example, are clusters of cells that make up all 3 major early cell types in the body: endoderm, mesoderm, and ectoderm. The iPSCs could also differentiate into neuronal cells and cardiac cells, which were observed to contract or beat. But this was all done in the petri dish. Could the cells differentiate in the body too? Indeed, when they injected iPSCs into mice, they developed teratomas, or tumors made up of different cell types. They were able to repeat these experiments in a couple of different adult human dermal fibroblast cells, a type of cell present in skin. So it worked. These 4 gene factors later became known as Yamanaka Factors and later earned Shinya Yamanaka the Nobel Prize. Although this was a big breakthrough in biotechnology, there's still a ways to go. For example, although iPSCs can form different cell types in the body, they could also form into cancerous cells. This process needs to be very well understood and carefully controlled in order for them to be used therapeutically. But this study opened the door for a whole lot more research into stem cell therapies, personalized medicine, mechanisms of cell biology, and more. What do you think? What is the future of stem cell research? Share in the comments. If you enjoyed this video, please share it with someone you think would enjoy learning about stem cells. Don't forget to subscribe to help us out, and remember,